This is part 2 of the walkthrough of the AQA GCSE Combined Science Biology 2023 Higher Tier Paper 2. Question 4. Sea lice are small animals that feed on the skin and blood of salmon fish. Figure 3 shows sea lice attached to the skin of a salmon. 4.1. What is the genus name of salmon? So this question is about classification. And what you need to remember are the different levels within the classification system, which was devised by Carl Linnaeus. Now they have many different names, so it's best to use something to remind you of the different names. So you can use a mnemonic, for example. And here is an example of a mnemonic. Do kids prefer candy over fresh green salad? So each of these letters will remind you of the names of those different levels of classification. So our G stands for genus, and the one below that is our species. Now these two make up what is known as the binomial name. So every organism has a binomial name. In this question now, our binomial name of our salmon is here. What we're looking for then is the genus, which will be the first name of the species. So we just need to state that first name. (4.2) Which domain are sea lice classified in? So our domain from our mnemonic now will be D. So it'll be the highest level. So we would expect many organisms to fall within a domain. But our examples of domains are only 3. And this is based on Carl Voigt's classification system. So 3 domains are archaea, bacteria, and eukaryota. Now a reminder that eukaryota is like eukaryote, and that is animals and plants, protists, fungi, organisms that contain cells with a nucleus. So our salmon and our sea lice are both examples of animals, so they would fall under the eukaryota domain. (4.3) Some salmon have genes that result in fewer sea lice attaching to the skin. Describe how fish farmers can selectively breed salmon that sea lice cannot attach to. So, there exists naturally salmon that have fewer sea lice attaching to the skin, but fish farmers can go through this process of selective breeding. We want to find out how we can produce salmon that have no sea lice attaching to them. So we have to recall the steps in selective breeding now. Now selective breeding is to do with reproduction, because that's what the word breed means. So we want to then have a look at the different fish that we already have and find those that are already containing these genes that result in fewer sea lice attaching to the skin. Now remember, if we're breeding, we need a male and a female, so it'd be best to state that first. Obtain a male and a female that have least number of sea lice attaching to their skin. Now, the next thing that we need to do is when we obtain that male and female fish, we need to breed them together. So they all produce offspring now. So what we're looking for with these offspring is we're looking for those offspring that will have the least amount of sea lice attached. Remember that when they're breeding, they're going to be passing on these genes. So we're expecting the offspring to eventually have these genes, which result in less sea lice attaching. So next thing they want to say then is select offspring that have the least amount of sea lice attached. We're going to breed them together. And we're going to keep on doing this for many generations. It doesn't just take one offspring cross for this to happen. It's going to happen over many, many generations. So they want to say they want to repeat this over many generations. And we're going to repeat this over many generations until all offspring have no sea lice attached. Remember, that's what the question is asking us. We want to get to a point where the sea lice cannot attach to the salmon. 4.4. Explain the advantages to salmon farmers of producing salmon that do not have sea lice attached to their skin. So ultimately, we're thinking about what's the purpose of doing the selective breeding that we described in 4.3. Well, why do farmers want to produce salmon? Well, it's probably going to be for sales, selling these fish for people to eat. So, a last point could be for fish farmers to make a better profit. Now the question is, they're still going to be having salmon even if they don't carry out this selective breeding. So how will they be able to generate a higher profit if these salmon are not having any sea lice attached? Well, if we go back to the beginning part of the question, it says that the sea lice are small animals that feed on the skin and blood of salmon fish. So every time that a sea lice attaches to the salmon, it's going to be taking the blood from the salmon. Now, what should be traveling in the blood of that fish? It really should be oxygen. So that can be something that we can think will be a point to include. Reminding ourselves that the sea lice will not be feeding on the blood of the salmon. Now, as we were describing just a moment ago, the blood should contain oxygen. How will that help the salmon? Well, the salmon will be able to carry out aerobic respiration. But if the salmon doesn't get enough oxygen because it doesn't have much blood that's being transported around, then it won't be able to carry out much respiration. And the point of respiration, whether it's an animal or plant, is to release energy. So a lack of this blood will mean a lack of oxygen, and then the salmon will release less energy. But if the sea lice is not feeding on the salmon, then the salmon will release more energy. And then what will be the benefit of that? Then more of the energy will be used for the growth of the salmon, so they will grow larger. And that's how we can link it to them making a greater profit. (4.5) Explain the disadvantage of selectively breeding salmon. Do not refer to cost or to time in your answer. So what's the issue with taking two fish, breeding them together, waiting for their offspring to grow, and then breeding the offspring together and repeating this over many generations? Well, if we were to draw it out, here are our two parent fish, male and female, and we breed them together, and we get many different offspring. And then remember that we're looking for the offspring that are displaying this desired characteristic the best. That's what selective breeding is. So, which ones have the least amount of sea lice attached to it? So imagine that it's this one and this one. And then we breed those together and then keep repeating that for many generations. Well, imagine that this sea lice that we chose here had a disease. And then we crossed it with this other sea lice here. Then that would mean that the genes that contribute to this disease would get passed on to the offspring. So we would expect some offspring there to have that disease. Now imagine that these offspring that have this disease are also showing the least amount of sea lice attached to them. We would definitely want to select those two. So what's happening then is this disease is also getting passed on as well as this desired characteristic. So eventually then, if we get to this point where all of our offspring are showing this desired characteristic, that would then mean that all of the offspring are showing or having this disorder. And that is a big problem. So the key point then is that because the fish at the end of our selective breeding process are going to be genetically similar specifically, then they're all going to be more likely to develop this inherited disorder. So all fish have many genes that are similar, because there will still be some variation, and then they're more likely to develop inherited disorders. Question 5. In vitro fertilization can be used to treat infertility. Which hormones are given to women having IVF treatment? Well, if a woman is infertile, one of the main issues is that they cannot produce eggs. So if they cannot produce eggs, we have to remember our normal situation for women that can produce eggs. What is the reason why they can produce eggs? It's because they have certain hormones that are being produced, and the hormone that is going to help mature eggs is going to be FSH. So what we're looking for then is an answer that is containing FSH, which is our first two. So we know that our last one can't be correct. But even though some women can produce eggs, they might not be able to release the eggs. There may be a problem with the fallopian tube, or there might be a problem with the ovaries itself. So we want to try and give the woman a hormone that allows the eggs to be released. So the hormone that allows the egg to be released is going to be LH. So that would mean that our first box would need to be ticked for the correct answer. 5.2. Name the target organ of the hormones used for IVF. Well, we know that hormones travel in the blood. But where do we want these hormones to go? Well, we want them to go to the place where the eggs are going to be developed and released. And that's going to be the ovaries. 5.3. Describe why microscopes are needed in the process of IVF. Well, what are microscopes there for? It allows us to see things that our naked eye cannot see. So, in this case of IVF, what would we want to see that our eyes cannot allow us to view? Well, it's going to be the eggs, because the egg cells are quite small in comparison to other things that we can see with our eyes. But in this case of IVF, what would we want to obtain the eggs for? We want to obtain the eggs so that we can check to see if these eggs are healthy. So that could be one point there, to check the eggs to see if they're healthy. Remember what's going to happen once we check these eggs and obtain them. We're then going to fertilize them with sperm cells. Remember that this happens in a petri dish. That's why it's called in vitro fertilization. So our egg cells and our sperm cells would fertilize, and this would be done outside of the body. So the other reason that we could use a microscope is to also ensure that the injection of the sperm into the egg is done properly. .4. Describe how the hormones given to women during IVF treatment interact with other hormones to prepare the body for pregnancy. Right. So interact, what does this mean? This means that one hormone, so let's use an example, hormone 1, can actually signal to cause hormone 2 to either decrease or increase in its amount. And that's what's really involved in the menstrual cycle. So we just need to remember now, hormones during IVF, we've got to be mentioning FSH and we've got to be mentioning LH, because those are the hormones that are involved. But it's talking about preparing the body for pregnancy. So when we're discussing this, we really should be thinking about the thickness of the uterus lining, because that's where the fertilized egg, the embryo, is going to stay in the uterus and develop from there. If the uterus line is not thick, then it's not going to be suitable enough for the embryo to stay and develop. So the question is, which hormone is there to build up and maintain the thickness of the uterus lining? Well, those are second group of hormones in the menstrual cycle, and that's estrogen and progesterone. So we just need to make the links now, interaction between these hormones, FSH, LH, estrogen, progesterone. So the way it works is if there's an increase in FSH, this will cause an increase in estrogen levels. Now, what is the estrogen there to do? The estrogen is there to thicken the uterus lining, as we said. Now, LH, when that increases, that causes an increase in progesterone levels. And what does progesterone levels, when they're high, do? That maintains the thickness of the uterus lining. Now, remember our last thing now. What is the importance of the uterus lining being thick? Well, in IVF treatment, remember that the fertilization happens outside of the woman's body. So once that does happen, fertilization, then the embryos are implanted back into the woman. So the uterus lining is then going to be ready for the implantation of the embryo. So our 3 points here now will be what these hormones do. So we need to mention both estrogen and progesterone levels. They have to be mentioned. Remember that the FSH is one of the hormones and LH is another one of those hormones. You could either mention any of those, but what's going to happen as a result? The progesterone and the estrogen, they work together to keep this uterus lining thick. And then, once the uterus lining is thick, it's going to be ready for implantation of the embryo. Scientists studied women who had IVF treatment. Table 1 shows the results. So here's our table. The number of women that received IVF treatment is 450. The number of women who had successful IVF treatment is 135. Calculate the simplest ratio of the number of women who had successful IVF to the number of women who had unsuccessful IVF. Calculate the simplest ratio of the number of women who had successful IVF to the number of women who had unsuccessful IVF. Give the ratio in whole numbers. So from the table, they have given us the number of successful IVF women. So we know that that's 135 right now. But they've given us the total, and that's 450. So for us to find the unsuccessful, it would be 450 minus 135. So that would be 315. So we know that 315 women were unsuccessful. So the ratio is 135 successful to 315, which is unsuccessful. But we need to find the simplest ratio. Even though these are whole numbers, there must be something that we're able to divide both sides by, so we can simplify this. Now, if we were to divide both sides by 135, that would not give us a whole number. But what we can do is we can divide both sides by the total. And that total was 450. So dividing 135 by 450 and dividing 315 by 450, this will give us a value of 0.3 to 0.7. Now again, that is not whole numbers. But what this is telling us now, though, is that every time that we've got a proportion, successful will be 0.3, or 3 times, and unsuccessful will be 0.7, or 7 times. So what we're able to do then, because it's just going to be a total of 1, we know that there'll be 3 lots of successful to 7 lots of unsuccessful. .6. So just one factor that can affect the probability of a woman having a child as a result of IVF treatment. Well, what could really, when it says affect the probability, is realistically what's going to decrease the probability? Well, one thing that can decrease the probability of a woman having a child through IVF is their age. So as women get older, the chances that this IVF treatment is a success decreases. And this is because of women going through the menopause, which will affect hormones and structures to make this probability lower. .7. Give 2 arguments against the use of IVF treatment. Do not refer to cost or to religion in your answer. Well, we were just talking about age and how that can decrease the probability of having a child through IVF. Well, the problem with IVF itself is that it has a low success rate. So going through the trouble of having IVF treatment, it doesn't guarantee that you actually will end up with a child. And this IVF treatment is actually very stressful and a very emotional journey that people go through when they make this decision to do this. So that is actually another point that you could say. It is a stress of going through this operation after operation after operation, surgery. These things can actually take a toll on the person's body. The other thing that happens in IVF is that once you have fertilized this egg cell with a sperm cell, it will produce many embryos. And these embryos are then planted into the mother. The problem with that then is that it can lead to multiple births. Now, multiple births sounds good, but on the mother itself, it is a bad issue. So this can actually increase the risks of problems, complications that occur to the mother, and could potentially lead to the mother dying. So multiple births are actually a risk to the mother. Question 6. The control of body temperature is an example of homeostasis. Give one other internal condition controlled by homeostasis. Do not refer to temperature in your answer. Well, they don't want us to refer to temperature because it was already been mentioned above. So we need another example. Well, homeostasis is the maintenance of a constant internal environment. So what inside our body do we want to keep the same so that it doesn't lead to issues with the human body? Well, there are a few. We just need one. One is glucose concentration in the blood. You want to keep that in a specific range. If it gets too high or too low, it can lead to problems. The common problem that happens when blood glucose concentration is too high, this is diabetes. The other thing that you could say is the levels of water in the blood. And that's regulated by the hormone ADH. But this is not part of your specification. Six point two. Explain why the control of body temperature is important. So why do we want to keep body temperature at a certain level? For information, the body temperature, internal body temperature, is 37 degrees. Well, we've got to think about what temperature could affect in our body. Well, temperature affects enzymes. So, having the temperature too high could lead to the enzyme being denatured, which is a change in the active site. So we want to keep this enzyme at a perfect condition so that it can quickly carry out its job, which is to catalyze reactions, break things down, for example. So what we can say then is enzymes, and this applies to all of them, have a temperature at which they work the best. And what we call that best is the optimum. If it goes away from that optimum, then the rate of the reaction is going to be lower. The body temperature of long-distance swimmers can change as the length of time swimming in cold water increases. Figure 4 shows how the body temperature of one swimmer changed in the first 4 hours of a long-distance swim. So looking at the graph now, our x-axis is telling us the time swimming in hours, our y-axis is the body temperature in degrees Celsius. What we can notice from the points is the body temperature decreases as the time in swimming passes. Six point three. Calculate the mean rate of body temperature decrease per hour in the first 4 hours of the swim. Well, our swim starts from this point and ends at this point. What we're trying to find out then is the decrease per hour in the first 4 hours of the swim. So we need to find out what this value is at 4 hours. But we can see here our first point, because it's over that time period, is at 37 degrees. So 37 is important for us right now. And then this value at 4 hours is going to be 35.5. We're trying to find the temperature decrease. We're going to subtract them. 1.5. Yeah. Our time period though is 4 hours. So trying to find the rate. So rate is going to be something, put x, over time. Well, in this case, our x is going to be body temperature decrease. So that's going to be 1.5. And we're going to do that over our time, which is 4 hours. So 1.5 divided by 4 is 0.375. .4. Hypothermia is a dangerously low body temperature. For this swimmer, a 5.5% decrease in body temperature from the start of the swim will cause hypothermia. Determine the body temperature at which the swimmer will start to have hypothermia. Give your answer to two significant figures. So what we've got to find out first then is, what is this 5.5% decrease going to look like on our graph? Well, for us to find this out, we need to do 5.5% of our starting value, because it does say a temperature decrease from the start of the swim. So our start is 37 degrees. So what we're able to do then is find this 5.5 by doing just a short calculation. And that's going to be 5.5 divided by 100 times 37. And that will equal 2.035. What we're then going to do is find out what's that decrease now by just subtracting. I'm going to take our 37 value, I'm going to subtract that 2.035, and then we should come out with an answer of 34.965. But it's asking us to give our answer to two significant figures. So two significant figures is going to be 35, because the number after the decimal place is 5 or more. A decrease in body temperature causes the adrenal glands and the thyroid gland to be stimulated. 6.5. Which gland secretes hormones to stimulate the adrenal glands? Well, our adrenal glands are situated just above our kidneys. A certain gland is going to secrete hormones to go to that location. Those hormones are going to be found in the blood, well, it's going to be a gland that's going to be important in controlling other glands. And that's going to be the master gland, but the actual name for that is the pituitary gland. 6.6. Explain the role of adrenal glands in responding to a decrease in the body temperature. 5 marks. So in this case, we need to discuss exactly what do the adrenal glands do. It says responding to a decrease in the body temperature. Well, a response to a decrease in the body temperature would be for the body temperature to increase again. So get back to around 37 degrees. Now, role of adrenal glands, which we can state straight away, it's in the name of the gland, adrenaline. So now what does adrenaline do? Many people associate adrenaline with a fight or flight response, and that's great, but we need to explain what's going on, so we need some detail here. So the release of adrenaline will actually cause an increase in the heart rate. Now, an increase in the heart rate should mean that there's more or faster blood flow, and the blood, just like in our previous question, is carrying oxygen. If it's this fight or flight response that we're talking about, it means that you'll be able to, or human or animal is going to be able to respond quicker to something. This means that their muscles in their body are going to be working faster. Now, if there's more oxygen and there's more glucose that's going to the muscles, this means that there's going to be more respiration, which is occurring. And respiration, when it occurs, it releases energy, and some of that energy can be in the form of heat. So this release of heat from respiration can be used to increase the body temperature. Six 6.7. Explain how the thyroid gland controls the response to a decrease in body temperature by negative feedback. Well, response to a decrease in the body temperature is going to then cause an increase in the body temperature as a result. But we're dealing with negative feedback now. So negative feedback is essentially the counteracting of a change that's occurred previously. But how's this linked to the thyroid gland? Well, again, just like the adrenal gland, it's in the name. So the thyroid gland releases the hormone thyroxine. Now, what the thyroxine is there to do is it's supposed to regulate the metabolic rate of the reactions that's occurring in the organism. But in this case, there's a decrease in the body temperature, and we want to respond to that by increasing the body temperature. So what we really want then is we want an increase in the metabolic rate, specifically, for example, respiration. So let's state the job of the thyroxine first, and then we can talk about what that does afterwards. Now, this increase in metabolic rate, that could be respiration, for example. But what will the release of thyroxine and therefore the increase in metabolic rate mean? That would then mean that there will be a decrease in the release of thyroxine due to this increase in the body temperature. And this is how negative feedback works.